it's a very busy, we're in a very busy moment in the work. And even with all this, you found a space to be here and with the Latin American community that is working in potato breeding and potato genetic resource. We're going to start now, and the question will be in the end, and the question will be in the chat or even with the open microphones, okay? So I'm going to share our presentation, and if you have any problem, just let me know, but I need you to talk, to say something, because now I'm not going to see anything else, only the presentation. Okay. Thank you, Hannele. Welcome to ALAP meeting.
Okay, thank you so much, Caroline. It's my pleasure to be here. So let's hope that the presentation works. It's a pity that I could not share my screen, so I cannot control the presentation, but I'm sure it will work out fine. Thank you.
It's okay. I think I'm going to mute my microphone in just a moment.
Yeah, if you can put it on the presentation mode, then. Okay, that's good. I hope everybody can hear me well. Good. Okay, so like Carolina already said, this is the title of my presentation. So I'm going to talk about genomic tools that we are currently using at SIP. So I'm trying to tell a story, how we arrived to where we are at this moment. And I hope some of these tools will be useful for you, and we can afterwards put you in contact with these people who run the services and so on. And you can also always come back to me or the SIP team for more information if needed. So next slide, please. So this slide is kind of to set the stage a little bit of thinking, why would we be using molecular markers? So if you think screening for biotic stress resistance in potato is very labor intensive, and it also takes lots of resources. So lots of funds go into this kind of work. So here I'm presenting 3 important diseases that we work, that are important traits in our breeding program at SIP. So we have the potato leaf roll virus, potato virus Y, and late blight. So there are, of course, other diseases, but I took these examples because these are diseases that we had already advanced and developed molecular markers for. So PLRV is a very difficult disease to work with, because you have to use aphids or grafting or infector plants in these assays. You need more than 2 seasons for the evaluation or to get reliable results. Then you have to do serological assay to get the results, and so on. PVY is a little bit easier to handle because you can do mechanical inoculation, but still then you have to do the serological tests. Late blight, equally, you need several seasons to evaluate, and if you do it in the greenhouse or in the field, in some places you need to inoculate, and oftentimes when you evaluate the resistance level it's a visual estimate, and you have to do these evaluations many times, so it's a long-term process. And sometimes you do not have the right pathogen strains available, for example. So it would be really, really nice if there were molecular markers available for these traits, and that would make our life a lot easier. Next slide, please, Caroline. So for late blight resistance, we have a long history at SIP working on this disease, and we had done quite a lot of molecular characterizations in our breeding germplasm, and we already knew that there is a strong QTL in chromosome 9 that explains a large amount of the resistance. And we had done GWAS using the SolCap array and also GPS, and we had identified some SNP markers that associate with resistance, and that this resistance is effective in Peru in many different seasons and years, and then we also found that it was effective in China. So this was a good locus for us to develop further and start using in the breeding program. Next slide, please. So we set out to develop an in-house assay for this locus, and so we took this SolCap SNP marker, and we developed this primers that were flanking the SNP, and through this HRM assay we were able to then differentiate the resistant and susceptible genotypes in the breeding program. And HRM, so high resolution melting, is not it's a relatively high throughput system, and the cost was around $4,000 per sample in those days. Next slide, please. We also knew quite a lot about the PVY resistance in our breeding populations. That it was mainly based on in chromosome 11, and on the RY and DGENA gene. And so we had already developed or used markers that other people had developed in a gel-based assay, so this RYSC3 marker. And then at SIP, colleagues developed another high-resolution melting marker based on this M6 marker. And here you can see how that HRM assay can differentiate the susceptible and resistant allele. So another tool we had then that could be used in the breeding program. Next slide, please. PLRV resistance, there was also a lot of work that had been done at SIP by other colleagues that they had mapped PLRV resistance from Solanum, tuberosum, and DGENA, and in chromosome 5. So this was also potentially an area of interest, and we worked very hard with Elisa Mijovilovic to develop a marker for screening in this locus. And the next bullet point there, and actually the picture that here, the alignment shows very recent work that we have been doing with the Sainsbury labs. So Jonathan Jones and his lab have been working towards cloning the resistance gene in that locus. So it is. We found that the RL and DGENA is a homologue of the BS4 resistance gene. Next slide, please. So before we knew the sequence of this gene, we had already then developed these scar markers based on we did a lot of work with trying to identify find the correct primers, and we had the scar marker, and then we developed a multiplex scar marker with the RL antigena and the RY antigena. So here we had yet another tool that we could use in the breeding program. Okay, so at this point we had markers for all these different traits. So PLRV resistance, PVY resistance, and late blight resistance. All right, next slide, please. But what was happening in the breeding program? Do you think that the breeders were using these markers? Well, they were not. And so the question is like, okay, why were the breeders not using? And we started to ask a little bit of these questions. So is there a disconnection between the breeders and the molecular breeders? Could it be that it's a very long turnaround time that before the breeders get the results and they cannot use it for taking any decisions? Maybe the cost is too high, or maybe they don't really know how to interpret the results, and maybe these markers don't even work in the breeding program. So we also did this cost comparison, and this is based on one single plate of 96 samples. So the cost of how much the DNA extraction is, PCR reagents, and all these different kind of items and chemicals and whatever staff time, bench fee that you need to run this assay. So we came into this cost for this high resolution melting. The cost per plate is close to $700, and the scar marker was then a little bit more expensive. And the turnaround time, if you run only 96 samples, this could be accomplished in 4 days. So it doesn't really sound that bad. If you have very few samples, this is something you can do. Next slide, please. But then we were part of a project that was involved several CGIAR centers, and it was called the High Throughput Genotyping Project. This was Gates, Bill and Melinda Gates Foundation funded project where they wanted to really push the CG centers to move towards marker-assisted selection. And so we were designing a forward selection plan in 2017. And in that case, we had a plan that we would genotype. In this case, we would use the PVY marker, and that we would genotype all the seedlings from the crossing block of that year and discard based on marker, and we would expect that about 50% of those seedlings would be then selected based on this marker. So if we look at the amount of samples that are here, 30,000 samples, this is not something that anybody can do in gel-based assays or even the high-resolution melting. It's going to take too long time. So next slide, please. So the challenge here was then to optimize and scale up the existing genotyping technologies for accelerated breeding while maintaining low cost. So in this project, then everybody who had some trait markers from the CG centers, we got the opportunity to convert them into this CASP system that is very high throughput. We had to set up a protocol for sampling and processing those samples, and a data management system and process how to return the results to the breeders. So what happened then in this project was that the CG centers, they negotiated a shared service agreement for the CASP genotyping platform at a private sector company called Intertech. And because all the CG centers were sending their samples or committing to send their samples into that same company, they were able to press down the price in such level that it was a lot more affordable. And the service system that they developed, it included DNA extraction and genotype, and they committed to a very quick turnaround time. So it would be between 10 to 15 days to receive the results. Next slide, please. Okay, so what we did then from SIP, we took the markers because we had the sequences available for the late blight resistance markers, the SNP markers, and the PVY resistance markers. And so in this table, you can see the SNP IDs from the original marker assays that we had. So the first one is a GPS marker. The next one is from the SolCap array. And the PVY resistance markers, they were just sequences that we had generated for the M6 marker for the PVY resistance. And then they were given these Intertech IDs. And here you can just see then that the gene or QTL that is based on the SNP, the two possible alleles for that SNP, and in which chromosome it is, the marker is located. And on the right-hand side, you can see the illustration of the results for the SNP for late blight resistance, that you can actually, even though Intertech returned the results as if these samples were diploids, you can see that there are different dosage. So all these different colors here indicate the dosage of the allele. The lower part of the slide gives the we recently published this study, so you can find that paper and learn more about this in more details. Next slide, please. So something that is important when we talk about the trait markers is that you need to do the assay verification, and it's because these markers are not diagnostic, so they are not designed in the gene sequences, but these are linked markers. And it's always context specific. So again, in that same paper, we are describing in what kind of materials we did it, but we calculated the false positive rate and the false negative rate and the assay power. So all of these markers, they worked fairly well in the SIP breeding populations. And it's also important to realize that none of these assays are perfect. So there will be always some level of error. But that error you will find also when you do phenotyping, or even if you do serological tests, there are always some errors. So sometimes people think that the molecular markers have to be perfect, but they really don't. You just have to decide what kind of a level of error you are ready to accept. Next slide, please. Okay, so this slide shows that there is a website at the Excellence in Breeding platform. So Excellence in Breeding platform is it's a platform for the CGIAR centers and also for the partners, and they provide help in many different aspects of modernization of breeding programs. And genotyping is one of those services that they provide. And here you can see all these different crops that have developed these CASP, low-density genotyping panels. And if you go on that website and you click on any of those pictures, then you can get the list of the trait markers that are available for those different crops. Next slide, please. So if you click that potato picture, it downloads an Excel file that has 2 sheets, and one of the sheets is this one. It's the potato CASP trait markers. These markers are publicly available, and you can see the first ones are the first 2 are the RY andigena marker, and then we have the RY stoloniferum and the late blight resistant markers. So until row 6, those markers come from SIP, except the RY stoloniferum. The others have been developed by our partners in the national program in India. So they got very interested in this opportunity, and they started developing their own markers, and they contacted Intertech, and they are using their services, and they also made their markers available for the community. So all these markers, if you are interested in, you can use them. So you can contract the service from Intertech. You can just tell them that I would like to use these set of specific markers, and you can send your samples, and you will get the results back. Of course, it costs, but it's a very affordable cost. Next slide, please. So the other sheet in that same Excel file is the list of quality control CASP markers that we have developed for potato. So that's a longer list. It is not shown here completely. So in the next slide, I'll explain a little bit more about this quality control set. So next slide, please. So we wanted to have a set of markers that could replace the microsatellite marker set that was used some years back for like a fingerprint for the variety identification, for example. Because we wanted to have this an easy assay that we could determine the true to type. So we always have a backup set where we multiply the tubers, and so the clean copy. And to go back to that clean copy, we want to be sure that it is still the same as the genotype that it goes through the evaluations in the field trials. So again, we used the SolCap markers, and we had genotyped our breeding populations, 206 clones from these 2 breeding populations, LTVR and LVHT. And we used some sets of filtering based on the minor allele frequency, and then diploidized these markers for easier processing, and tried also to identify 2 markers per chromosome. And then we validated this, and actually it is ready for routine use. Next slide, please. So this is another slide that the details you can find in that same paper that I was referring to earlier, about how we have used the QC assay. So we took samples from greenhouse, from different locations in the field, and we're trying to identify if the genotype that we had in the greenhouse when it had the same name, if it still maintained, if it was still the same in the field. And we actually did find quite a bit of errors. So I'm not going into the details of that table, but you can get more details in that paper. So, but with these 24 markers that we have, and using the tetraploid dosage, we are able to identify full SIBs. So we think that these 24 markers are sufficient amount for us to use in fingerprinting in our breeding program. Next slide, please. Okay, so this is just a slide from my colleague in the Excellence in Breeding, and which now belongs into the Breeding Resources Initiative in the new One CGIAR structure. So they are providing this low-density genotyping service using the CASP genotyping method, and that the primary application for this is the quality control that I just showed earlier, and the trait-specific markers that I also already described. So in CGIAR, these CASP markers are available for 18 crops and fish, and the targeted users are CGIAR centers, but also the national partners and advanced research center institutes as well. The cost, it depends on the number of markers to use, but initially when we were using, so we had 10 SNPs, then the cost was, it was about $3 per genotype. So so for one genotype, you would get results of 10 different SNPs, and that would cost $3. Again, it takes maximum up to 15 days until these results are back from the service provider. From the day that they receive the samples, they guarantee that within 15 days the results, you can get the results. So next slide, please. This is just to show some statistics, again from my colleague, about the usage of this system in the CG and national breeding programs in Africa in year 2020. So you can see the breakdown by different crops, cereals, pulses and roots, tubers and bananas. And you can see that there are lots of users for this system already. Next slide, please. And here we see the number of samples that were genotyped in Africa in 2021. You can see that there were not so many samples of potato in 2021. And on the right-hand side, you can see the number of markers that were used. So this year, today, the numbers are already different, but 2021 was COVID, and things were a little bit more restricted. So next slide, please. Okay, so that was the low density markers, so the trait markers and the quality control. But another important genotyping approach, or what we normally use, is genome-wide markers. Okay, so in the past, very long time ago, we used to use the SSR markers and COS markers for fingerprinting and genetic mapping at SIP. Then along came the Illumina SolCap SNP, and that we used for genetic mapping and GWAS. And this we did with the service provider. There are a couple of them. And we were also using the GPS markers for the same purpose as the SolCap. And in this case, this was already, I think this was in 2016. And at that time, the service provider was not able to call these markers with the allele dosage, so we had to do the bioinformatics ourselves. So in that next column there, I'm trying to make to describe the technical effort, what it takes to run those different markers. So if we run them, the SSR and COS, it's a little bit more technical effort because you do the DNA extraction and then you run the markers and a lot of work in the in-house lab. These SNP markers, so it was only a DNA extraction, but then there is quite a lot of data processing effort, particularly in the GBS platform. And then the turnaround time, how quickly you get the results, and yeah. And I did not get to the cost sample data point here, but that's okay. So next slide, please. This is just to show how complex it was in those days, or yeah, there are many steps if one has to call the genotypes from the GBS, so the genotyping by sequencing data. And so you really need to have somebody who is able to invest a lot of time in bioinformatics. It's a nice system, but it's very intensive, takes a lot of time to do this work. Next slide, please. So again, we wanted to have a system that is more user-friendly, and also that we don't need that many markers. So Excellence in Breeding platform was developing this mid-density platform for different crops. And at this point, we started to collaborate with Jeff Endelman at the Wisconsin. And the service provider for this genotyping was the Diversity Arrays technology in Australia, in collaboration with the Intertech, who did the DNA extractions. So again, we started with the SolCap SNP markers, and we were selecting these markers in 1 centimorgan bins per chromosome, and different kinds of filtering. We came down to 2,501 markers. So this DART tag, you can read the details of these technologies in the DART website, so it is a simple bioinformatics and relatively low cost, and any mapped marker can be included into this assay. Next slide, please. So we have started to use these markers. We've been testing them, how good they would be working in genomic selection. So we've done some cross-validation. We have a PhD student who was working with Mokhtar Kante, who was here I think, also joined this meeting. And so Joao, and also with Jeff Endelman. So he has done nice work with the phenotypic data from ELISA's program in Peru, and looking at the prediction accuracies for different traits. And if you can see here, all the prediction accuracies are very high for all of the traits. So we think that this is a very good platform, and we really want to start using it routinely in genomic selection in the potato breeding. Next slide, please. So the DART tag, we have selected these markers, but in DART company, they have also other technologies available. So they have this DART-seq system, which is basically very similar to genotyping by sequencing. And especially they have the low density platform that is very affordable. And sometimes, we were thinking that this platform could maybe be just as good as the DART tag. So we ran this test, and so calling this testing the markers from the DART tag system, but also from the low density DART-seq and the high density DART-seq. So starting from these different marker numbers, Joao, again, he was looking at how long time it takes to call these markers. And you can see that there's a significant difference. So from the DART tag, it's only 1.5 hours, while the high density, it can take up to 2 days. The final number of markers that you can then use, it depends on the filtering and different kind of things. But so then now we had all these different sets of markers. Next slide, please. This slide shows the distribution of these different markers from all these different DART platforms along the chromosomes. And you can see that they are fairly distributed in a fairly similar way. Next slide, please. He was also looking at the prediction accuracies for all these different platforms and the different kind of filterings that he was doing. So this is work that has not been yet published, but the manuscript is going to be published pretty soon. So I'm not going to go into the very deep details, but this genomic prediction here, the trait, is for tuber weight. And you can see that this line has moved a little bit, but you can see that the best prediction accuracies are always for the DART tag system. Next slide, please. So that was for the tuber weight. And here we have another content. DART tag mark form better than the DART-seq low density or high density markers. Next slide, please. And Joao even managed to get some significant SNPs in the GWAS with this marker system. So he found significant SNPs for the senescence and the drought treatment. So it's because these are big effect loci that they could be detected with these markers, that it's quite a low density, so it's not really suitable for GWAS. Next slide, please. Okay, so again, this mid-density genotyping service is available from the Excellence in Breeding and the Breeding Resources. So the platform that they use is the DART tag genotyping method, and the primary application intended here is for genomic selection, but it can also be used for diversity studies and DNA fingerprinting. They have 9 DART tag panels validated, and again, the same users, the CGIAR and national programs. And here the cost is $11 per sample, and the turnaround time again is maximum 15 days. Next slide, please. Okay, here is another summary of showing all these crops that have developed these dark text panels available, so you can see rice, potato, wheat, maize, and others. And so the vendor is the dart, but in most crops, just like in potato, there is it's a two-step procedure. So the samples are first sent to Intertech in Australia because they are allowed to receive leaf material. They do the DNA extraction and then send the plates to DART, and who does the genotyping. Next slide, please. Okay, so this slide is to show the genotyping workflow. So the users will fill in the form and confirm the samples in the field. So there is a special order form that we have to fill and send to the vendor after we have collected the samples. Then we complete the phytosanitary certificate and the vendor's form and the commercial invoice. We ship the samples to the vendor, and when the vendor gets the samples, they do the DNA extraction, sequencing, and genotyping in 15 days and return the results to the whoever submitted the samples. Next slide, please. Okay, so then just a few words about how important it is to have also a database system if one is working with such high big volumes of data. So it's really important to be organized. And so for the roots and tubers and banana crops, there is a database that is shared. It's called a breed base. And for each crop, they have their own specific details. And so potato base is available for any users that are interested and want to have their own instance installed, and potato base can receive genotypic data. It's also important to develop these SOPs and protocols for data management. We have done that already for sweet potato, and we are working on it for potato. So it's really important to have these data workflows and also digital data collection, and to use barcode labels, have the naming conventions and everything well defined. Then there also, of course, has to be a data curation steps and so on. But it's really quite difficult to manage this kind of data when it comes back only on in Excel and in your own computer. So I'm right now struggling to pull out all the old historic data and get it into potato base. So if you are interested in starting to use this kind of genotyping services, I definitely recommend to get some kind of a database to manage those results. Next slide, please. Okay, so just some lessons here. So I think, like you saw from those examples in the beginning, that at SIP we had developed all kind of molecular markers, but nobody was using them. So really the molecular team has to be very, very much integrated with the breeders. And so because they need to have buy-in, otherwise the markers are never going to be used. So what we did at SIP, because we had a separate budget for genotyping, so we paid for the genotyping for the breeders, and when they were then convinced that the results were okay, they were willing to start using the system. It's also very important to have clear roles and responsibilities within the teams and make sure that it's well defined who collects the sample. To be a sampling strategy in place and protocols of standard operational procedures, some scripts for data analysis and to support the results interpretation. Next, please. And the data management. So if there is no database, it's really important to organize all files, to have the genotyping order forms, the original results files, and the processed files and everything. And it's very important to be consistent with the sample names and the trial names. And if any large-scale genotyping happens, database is really, really essential for that. Next slide, please. Okay, so in our case, what we want to do is to get the potato base fully operational and running with all the genotypic data. And we also want to have actually that the vendors can directly return the data directly into the potato base instead of sending them to the breeders and that they would have to upload the data again. We would also like to generate the order forms from the potato base directly. And some of these logistics of when we are going to use the QC markers and so on. We are also working on genome assembly for better marker development. So we are working on sequencing some SIP potato lines. And also we want to add all the markers, the trait markers, into the mid-density marker set and also the QC markers, so that we can also have only one single platform or get all the data from one single run if we want to. Next slide, please. Okay, this is just a slide with some of the resources that might be useful for you. Next slide. That's the final slide. So I just want to thank the partners and the service providers. Thank you very much. Happy to have any questions.
Thank you, Hannele. It was very good to hear and to see your experience and to share with us. I'm just going to see if there is any question here in the chat. Let's see. Guilherme, please, could you please make a question?
Yes. Hi. Hello, Hannele. Very nice talk. Thank you. I have actually two questions, kind of related, about the cost markers. So do you know the error rate from the SCAR and HRM techniques? I'm guessing they could be even higher due to the lack of dosage information. I'm not sure if you can get it from HRM. And a follow-up question is, how many markers do you consider enough for selection? Consider that could have errors, right? Like you run 2 or 3 markers, then you see the overlaps, or 1 would be enough. I'm guessing the cost wouldn't be that great by adding 2 or 3 markers, but I'm just trying to figure out the way you're doing. Thank you.
Hi, Guy, nice to see you, and thanks for the questions. So we never did look at the error rate in the SCAR markers or the HRM. The HRM can get the dosage also, so we were able to see that. But I don't know the error rate. But I expect it can be bigger than from the SNPs, indeed. Yeah. So about the next question, so the value of markers, I think it depends on the traits. So even a single marker can have a big value. So for the PVY resistance, for example, I mean, so we genotyped, in this case, we always got the results of 10 SNPs, but for some breeding materials, we would be only interested in one marker. So it really depends, I think. Intertech is trying to set up these protocols in a way that you can have a set of SNPs, and then the price depends on that. So yeah, the 10 marker point was where they could give the price around $3 per sample. But if you added more markers, then it would get progressively a little bit more expensive. Did I answer your question, or did you miss something?
Yeah, okay. That was great, actually. If I may, I have actually another question, and that's about the very high predictive ability of the genomic selection work by João in Moctar. I'm not sure if that has to do with the F1, with the fact that it was based on F1 populations, like 3, and then the I guess the number of alleles segregating would be lower than, in comparison with a breeding population. Did you guys investigate that by any chance or no?
I have to give this question to Moctar. Is he still around? He could respond.
Yeah, do you hear me?
Yes.
Yeah, I mean, that's one explanation, and the other one is also that, as Hannele said, it's a cross-validation, so it's not really genomic selection prediction accuracy. It's and actually when you do cross-validation, you really have very high prediction accuracies, but then when you really predict what's in your breeding population, actually it really decreases. Maybe we would expect 0.4 at most of prediction accuracy.
Right. Awesome. Thank you, Moctar and Hannele.
Yeah.
Thank you. We have a question in the chat from Napersa. I can read. She's an analyst at the Molecular Biology Laboratory here at Embrapa. And in fact, we need to buy a new equipment for SSR genotyping and other markers. What type and brand of equipment are you using in SIP for SSR genotype? What do you recommend? In fact, we have here the LICO, the same that we used to have in SIP in the past, and we use the 24, the kit for 24 SSR markers for quality control. We don't know if you are still using some in-house genotyping, or you are sending everything to do at EAB.
Yes, so we are not using any in-house genotyping anymore, and particularly we are trying to move away from all old technologies. So SSRs are not really. I remember during this high throughput genotyping project, the donor was like, he was very upset if people were using SSRs. So he was really trying to push everybody move away from that technology, because it's not that high throughput, really. I mean, I think it's not very cost effective. So I would really recommend to try to move into this service, like the CASP. You could try and see if it works for you, and even develop own markers for yourself. So everybody can use this system. I think in some cases it might be cost effective to run the marker work in your own lab, but in most cases it's just too expensive. At least in SIP, we cannot afford to do that.
Okay, thank you. Natasha, it's okay, the answer? Okay, so we have a question from Elisa. Yes.
Sí, Elisa, gracias por la pregunta. Yo creo que podríamos organizar unos eventos de entrenamiento como se muestrea, por ejemplo, y cómo se envía las muestras. Luego, yo creo que no podemos a través de SIP coordinar el envío de las muestras, sino que cada institución debería contactarse con Intertech, por ejemplo, o DART, pero podemos enviar, ponernos a ustedes en contacto con estas personas. Eso ya hemos hecho con varios. Tal vez Guy puede comentar porque él también está trabajando con esas plataformas.
Sure. I'm gonna speak in English. My Spanish is not so good, but we are actually doing it. But the main point of contact is actually EIB, because EIB says that if it's a public breeding program, we can use their service and their pricing instead of doing it through Intertech directly. Like, of course, we can do it through Intertech and DART, but if we do it through EIB, that would be much easier. So they take care. They look at the paperwork, they look at the spreadsheets, they look at everything, they make sure everything's going to work. And then we pay EIB actually instead of Intertech or DART directly, right? Because of that agreement. So as far as I know, if it's a public breeding program, we can use it. We don't need to do it through Intertech or DART directly unless you want to, right?
Okay, gracias. I have a question, Leader. I was in the workflow of the genotyping service. We have the phytosanitary certificate and the invoice, but I didn't see the part of the legal part, the transfer agreement. Because, for example, here at Embrapa, we need to have an agreement that we are sending the samples. So do you have any standard made already, or how does it work? You can talk about.
Yes, I'm trying to remember. There is a form. Mokhtar, do you remember the details of that? There is a kind of an SMTA, right? So what we are always sending is leaf discs, dried leaf discs, and they go into a destructive service. So therefore, there is no, they are not going to use that for anything, and they return the data to you. So from that perspective, it's not required.
Yeah, and just to add something, also they can keep the samples. For example, once they receive and process the samples, they would keep them for 3 additional months, just in case you want to verify something, you want to rerun some SNPs on the same samples. And after that period, they destruct all the samples unless the user requests to keep them. So we never felt we really have to worry about that part.
Okay, thank you. Yes, we have a specific agreement here at Embrapa that we use when we send samples like DNA or NIC discs, and it only says this, that the results will come back to the institution, that it cannot be used, and also that the sample will be destroyed. But we need that the other part needs to sign this. So that's only the point that maybe would be interesting to have in this workflow to be either for the institutions, like we have this problem here, that we cannot send anything without this agreement. So just maybe it would be interesting. Thank you for the information.
Yeah, I think it can be solved, all of it, because they really want to work with national partners. And I think there are some cases things can be more difficult than in others. So they are really very service oriented. So I'm sure it can be worked out.
So any other questions? I have one more question, sorry. It's regarding the late blight resistance, the markers. Did you validate in other populations background, because or only in the sick populations? Can you talk about this?
No, we have not done that, but there is a quite recent paper from the Indian group, and they validated these SNP markers. Now, I do not remember the results, but I'll find that paper for you and I can share it. So, but yeah, I mean, you really have to check in your background because they will not work everywhere, those markers. Yeah.
Okay, thank you. So, any other question or comment? Hannele, I think it was really good to have this moment of these questions. Everybody, all the doubts are common, and we had a very good moment to know about this platform. That, as I said to you before, I knew this about Guy. It was the first time that I knew that how it was working. So, and I think that other persons here, maybe it's the first time that have the opportunity to see this. And just like maybe my final question is, do you know if there is any initiative at EIB, or maybe Guilherme can also answer this, to have more national breeding programs included in this platform? It means like any to make it easy. It means like financial resources, because we know that mainly here in Latin America we have a lot of problems, financial problems. And another question is that I saw that at EIB there is a model that is for the data analysis. So can you talk about this, because you said, and this is even with the data sector, it's also a problem when a breeder receives the results to make this interpretation. If you can talk about your experience. Thank you.
Yes. So in CGIAR, most of the donors are really interested in funding everything that has to do with Africa. So there is not that many resources coming to Latin America, but we can still benefit from in different ways. And for example, one of the things that we want to do and request from the Breeding Resources Initiative is to give trainings in different areas, have specific trainings for people in Latin America and other trainings in Africa and Asia and so on. So I think this year we probably will be able to benefit from some of those services and also include the Latin American national partners in some of these trainings. But we could also think of something coordinated from SIP and maybe through this ALAP or something like that. It can be also virtual things, that those are much more easier to organize because it doesn't require very much resources, right? So, yeah. But I even asked, so last week I was in a meeting where this genotyping coordinator from Excellence in Breeding was, and I asked if she would be also providing training in Latin America, and she said yes. So maybe we can organize something like that.
Yeah, it would be very good to have this for Latin America also. And well, we will have the ALAP, the Congresso in Chile in March, and some breeders of several countries in South America will be there. Maybe it's a good place to start something, and these virtual courses for sure are really good. You can have a lot of people in a part of this. So very nice. So thank you, Hannele, and also thank you everybody that is here, collaborate and listen. It was a very, very good moment that we had here. And again, Hannele, thank you very much to share this experience and to show all these interesting results and experiences with us. Thank you.
Thank you so much, Caroline and Elisa. Thanks for the invite. It was my pleasure. And let's keep in touch. Adios.
Gracias.
Gracias.